So we continue with the control of the cell cycle. Many scientists study the control of the cell cycle, the different checkpoints, because controlling cell cycle, that means you can control cell division. When you are able to control cell division, you can potentially control the cancer cell. So I want to emphasize this one term, tumor suppressor gene. As the name suggested, this gene suppresses the tumor in an inhibitory pathway. If the tumor suppressor gene is mutated, or if it for any reason doesn't work, then the end result may be unregulated cell division. Unregulated cell division, what does it ring a bell to in your head? They are basically cancer. So here is an extremely simplified diagram for tumor suppressor gene stuff. So you see that some kind of stimulating growth factor will bind to the integral protein on the cell membrane, and then this protein will trigger a signal transduction cascade, maybe the cyclic AMP pathway, and then the signaling molecule can go into the nucleus through the nuclear pore, and then activate the ribosome to sit with the nucleus, so that the ribosome can start to translate into protein, to translate the DNA sequence into protein. Another way to suppress the tumor gene is that the tumor suppressor gene is mutated. So what happens is that, okay, you also have the growth factor, and then you have signaling pathway, and then you have the transcription factor. And because it is a wrong inhibiting growth factor, then what happens is that the protein that is unable to inhibit the cell cycle or promote apoptosis, they will end up making some useless protein. So this is what I meant. Okay, in the cell you have the what's that called? So the inhibiting growth factor, when it binds the receptor will start a signaling cascade that will activate a transcription factor. This transcription factor will sit on the DNA so that you will inhibit the cell from replicating its own DNA. So I want to tell people that, okay, the DNA is arranged in different level, organization level. The lowest, or we call it the simplest organization level, is that DNA, they are freely floating in the inside the nucleus with some kind of what's that called? Is it a medicine? Or, no. Anyway, this DNA will be freely floating inside the nucleus, and then also you will surround some of the so we call it packaging protein. The packaging protein is called histamine. Histamine, after it is wrapped around the histamine, then you will further coil up like this to make a foam cord. So the replication of DNA, of course, occurs in the S phase. it will cause, it will make two sister chromatid. One is the original one, the other one is the translated one. So again, it repeats itself. DNA replicated in the S phase, and then after S phase, you will see two sister chromatid. And this makes you even more confused. Centromere. Centromere. Centromere refer to the ball-like structure here in the center. But nowadays, many people abuse this term. For example, they say that, oh, I'm going to South Africa, no, it's okay. We'll talk about the three different centromere, centrosome, and centriole. This is called karyotype, meaning that it is ideally you should do this before you want to have baby. The reason is that the doctor can screen through your chromatids, make sure that your kid does not have Down syndrome. So the definition of karyotype is given here. The display of all chromosomes in a somatic cell is called the karyotype. And so remember that the number of chromosomes does not indicate the total number of genes. Gene is a relatively smaller subunit you can find in the chromosome. For somatic cells, we have two copies of the chromosome, meaning that we have one copy from our father, we have another copy from our mother. We call it homologous pair. Homologous pair. We have 46 chromosomes, but when they pair, we only have 23 pair of chromosomes. So this is the brief overview of the mitosis cycle. At first, it starts with interphase, and then after interphase, when people like her, then she becomes prophase. And then after that, the metaphase. After metaphase, you have anaphase, and at the end, you have telophase. So what about mitosis? What can mitosis break down? You can break down phosphase, metaphase, anaphase, and telophase. So the main rule of mitosis again is to separate sister chromatids so that the cell will have each of their daughter cells. Now let's take a look at interphase one by one. So it looks like this interphase, but if you look at it under the light microscope, you see that a cell that is not as clear as this one, clear meaning black and white, but it has a lot of white metal finishing on the surface of the interphase. In the interphase, the DNA there in the chromatin form is like spaghetti, and you have two centrosomes, double nuclear membrane and double plasma membrane. Now in the early prophase, so how can you tell the differences between a prophase and interphase? Is you can look at it by their color of the eyes. If the color of the eyes is very conspicuous, then it is interphase. If the colors of the prophase, if the colors of the eyes of the prophase is relatively lighter blue, then they are not in the lighter blue. That means it can be prophase. There are also other characteristics. Is that in the prophase you can start seeing chromosome instead of chromatids. Also, what happens is that during the interphase, the two centrosomes move to move perpendicular against each other. Okay.